Hello friends, welcome back. And friends, as you know that in our series on cell biology, we have discovered and studied a lot. So continuing our series on cell biology, today we are going to talk on one of the another interesting topics, and the topic of today's discussion is second messengers in cell signaling. And for the discussion on the topic, we have with us in our studios Dr. Varunendra Singh Rawat. Dr. Varunendra Singh Rawat is Assistant Professor in Department of Zoology, and he is teaching in Hindu College, University of Delhi. Dr. Rawat is a prolific professor, and through him we always get in-depth knowledge on various topics. So let's welcome our guest, Dr. Varunendra Singh Rawat, and let's try to have a vivacious session once again. Hello sir, welcome to the lecture.
Thank you. So today I'll be talking about a very important aspect of cell signaling. We have been covering a lot on cell signaling because it's a very important process which ensures the survival of the organism. Because if the cells are not able to communicate with each other properly, there would be a disastrous consequences for the cells, for the tissue and the entire organism itself. So cell signaling by itself is a very important process, and there are a number of molecules which are involved in this particular process. As the name of this particular topic suggests, second messengers and their role in cell signaling, which implies that there must be first messenger. So the first messenger is basically the molecule which is released by a cell, and it is uptaken by another cell by the virtue of the presence of receptors on the second cell. So that is the first messenger. It brings in the message. Once the message has been conveyed to the particular cell, once the message has been received by the receptor on the cell, then the process of signal transduction starts. That is, this message, it leads to formation of certain other molecules which will start or initiate processes towards the downwards of the pathway. So the molecules which are generated by this particular process are termed as second messengers, and these are the molecules which will initiate the downward process or the processes which start from their end. So today I'll be talking about second messengers in detail, and the outline for this particular topic. The topics I'll be covering in this particular lecture would be basically it will be dealing with lipid based second messengers and ionic messengers, because I've already covered the CAMP and CGMP, which are very important second messengers. They have already been covered elsewhere. So I'll be covering basically the lipid based secondary messengers and ionic messengers in this particular topic, in this particular lecture. And the objectives for this particular lecture would be to talk about what different types of lipid based second messengers are there, and how actually they function, and how actually they help in transduction of signals. So starting with the basic thing that primary messengers come and they bind to the specific receptors on the surface of the cell, and they generate secondary messengers by activity of certain enzymes. So we already know CAMP and CGMP are involved in specific pathways like G-coupled protein receptors, and then there are certain other molecules which are dependent on lipids, which are formed by processing lipids present in the membrane. So this particular panel shows there are specific molecules like PIP3, PIP2, DAG, IP3, which are derived from lipids by processing of the lipids present on the membrane. Then there are molecules like CAMP and CGMP which are also generated primarily by GPCR, that is G protein coupled receptors, which have been covered elsewhere. So I'll be talking about these molecules in detail, like PIP3, DAG, IP3, what exactly is their role and how they function in the cell as second messenger. So their building blocks are basically phosphoinositide. These phosphoinositides are phosphorylated phosphatidylinosytols, which are usually found in the inner leaflet of the plasma membrane, which is also termed as cytoplasmic leaflet or cytoplasmic side of the cell membrane. The simplest molecule is termed as myoinositol, which has 6 hydroxyl groups, and 3 of these 6 hydroxyl groups can be phosphorylated. So this particular myoinositol, it can be phosphorylated at 3 different positions simultaneously or one position at a time can be phosphorylated, one hydroxyl group can be phosphorylated. The mammals produce 7 different types of phosphoinositides, each of which is found in a different domain of the membrane. That is, they are present at different sites in the membrane, and each of which can be differentiated by the location of the phosphorylation of the inositol ring. That is, these 7 species, they have different location wherein the hydroxyl group is getting phosphorylated. So there are at least 7 species of these molecules present in the membrane of the cytoplasmic side of the membrane of the mammal. These phosphoinositides are involved in a number of activities, like they can regulate a large number of cellular events like cell signaling. They are also involved in membrane transport and regulation, membrane transport regulation, cytokinesis, and as well as organelle distinction. They can do this directly by binding to the proteins or by activating certain proteins which lead to a structural change in the protein, and they can also lead to change in the process by mediating a number of signaling pathways. So these are the ways in which phosphoinositides mediate in a number of pathways occurring in the cell. So talking about lipid derived second messengers, the discovery came in late 1960s to 1970s that these lipids, which were earlier thought to be a part of membrane only, that they did not play any other part apart from providing structural framework to the membrane. The discovery came later on, and it was found that number of extracellular signals which were perceived by the cell, they promoted the hydrolysis of phosphoinositides in the cell. And once these phosphoinositides were hydrolyzed, it led to generation of certain molecules which act as second messengers. So DAG was identified, diacyl glycerol was identified as the major second messengers by year 1970s, and a scientist named as Yasutomi Nishizuka purified the enzyme protein kinase C, PKC, and showed that it is the target for DAG. That means DAG once formed, it acted on this particular kinase, protein kinase C, which then acted on the other proteins which were taking part in that particular signaling pathway. A number of, as I've already told, a number of extracellular signal can trigger the hydrolysis of the phosphoinositides or the lipids which are present in the membrane, in the inner membrane of the inner leaflet of the membrane, and these can be categorized as glycerolipids whose hydrophobic backbone is made by DAG. And the example of such glycerolipids are phosphatidylinositol 4,5-bis phosphate, the short acronym for which is PIP2, and the second class is sphingolipids whose hydrophobic backbone is ceramide, and the example of which is sphingomyelin. This panel particularly shows different types of enzymes which act at different positions to generate different types of second messengers, like PLC, phospholipase C, phospholipase A1, phospholipase A2, which act at different position of these phosphoinositides to generate specific second messengers. Now, binding of ligand to receptors which couple to lipid modifying enzymes results in the activation of enzyme which hydrolyze specific acyl chains or polar head groups from each lipid class to generate lipid second messengers. Like in this particular panel, there is a head group which can be cleaved off, or there are fatty acid chains which can be cleaved off, and these polar head groups and fatty acyl chains, they themselves act as second messengers. So hydrolysis of acyl chain can generate either lysophospholipids, example of which are lysophosphatidylcholine and lysophosphatidic acid, which are generated through hydrolysis, which is catalyzed by phospholipase A2, which is PLA2 as shown in the earlier panel, or they could be lysosphingolipids, example are sphingosine, which are generated through hydrolysis catalyzed by ceramidases. And then also are generated free fatty acid chains like arachidonic acid. So these are some of the messengers which are generated by action of specific enzymes acting on these phospholipids. Now, hydrolysis of polar head groups from the glycerophospholipids by phospholipase D and phospholipase C results in generation of phosphatidic acid and DAG respectively. So when PLC acts on the phospholipid and the head group is cleaved off, it leads to formation of DAG, and when phospholipase D acts on the phospholipid and cleaves off the head group, it leads to formation of phosphatidic acid. The polar head group which is released by following the hydrolysis of PIP2 is termed as IP3, which acts as a second messenger. This IP3 can also be further phosphorylated to form polyphosphoinositols that serve as additional second messengers. So here we can see that IP3, which is generated as a head group after the cleaving of the head group, it acts on itself as a second messenger, but different groups can also be phosphorylated to generate more second messengers through this simple molecule of IP3. Apart from that, PIP2 can be phosphorylated itself to produce another messenger, key messenger, which is PIP3, which is a very important messenger involved in cell signaling various types of cells. This particular panel shows how a particular glycerophospholipid can be cleaved in a variety of ways to generate a large number of second messengers. As we see, when phospholipase A2 acts on the glycerophospholipid, it can give rise to lysophospholipid. When it is acted upon by phospholipase C, it leads to formation of DAG or diacyl glycerol. When it is acted upon by phospholipase D, it leads to formation of phosphatidic acid. And other messengers like arachidonic acid and inositol 1,4,5-trisphosphate or IP3 are also generated by cleaving of this particular glycerophospholipid molecule. As you can see, IP3 is a head group and arachidonic acid is a free fatty acid chain. The hydrolysis of the polar head group from sphingomyelin, on the other hand, it's catalyzed by sphingomyelinase and it produces ceramide, which is another molecule which acts as a second messenger. Further hydrolysis of the acyl chain by ceramidase produces sphingosine, and its subsequent phosphorylation by sphingosine kinase produces sphingosine 1-phosphate. So we can see here, hydrolysis of sphingomyelin leads to formation of ceramide, and further action by the enzyme ceramidase produces another second messenger, which is sphingosine. When this sphingosine is phosphorylated, it leads to production of another messenger, which is sphingosine 1-phosphate, and each of these have separate role or different pathway to act upon, which we will see on later on. So this particular panel shows how sphingomyelin, when it is acted upon by sphingomyelinase, it leads to production of ceramide. Ceramide, when acted upon by ceramidase, it leads to production of sphingosine. Sphingosine, when it is acted upon by sphingosine kinase, it leads to production of sphingosine 1-phosphate, which is another type of second messenger. Sphingosine 1-phosphate can be converted back to sphingosine by removal of the phosphate group by enzyme sphingosine phosphatase. So all these molecules they can act as second messengers in the cell. Now, lipid second messengers which are able to retain two acyl chains, for the example DAG and phosphatidic acid, they remain associated with membrane. That means these second messengers do not leave the membrane, they are bound to membrane, and they remain bound to the membrane, and therein only they activate certain other protein. Whereas lipid messengers which retain only one acyl chain, like lysophosphatidic acid and sphingosine, they are allowed to dissociate from the membrane and they serve as soluble second messenger. IP3 is also a molecule which does not have any acyl chain bound to it, so it also serves as a soluble messenger, soluble second messenger. So here we see when there is a signal and there is activation of enzyme like PLC beta which acts upon PIP2, this PIP2 is broken down into IP3 and DAG, two molecules, DAG which is the second messenger by itself and IP3 which is the soluble second messenger. These two molecules have different mode of action, which I will be talking about later on, but what exactly I mean to say is that by when a message is received by the cell, it can lead to activation of enzyme like PLC phospholipase C beta, which then acts upon a specific phospholipids PIP2, which can be broken down into two secondary messengers, each of which have different activity and different location in the cell. So when signal from GPCR and RTK are transmitted, they result in activation of PLC, which was shown in the earlier panel, and this cleaves the phospholipid to generate two specific molecules, DAG and IP3. In the case of GPCR signaling, lipid hydrolysis is mediated by binding of G proteins, specifically GQ protein, to phospholipase. That means G proteins are bound to phospholipase PLCB, whereas in the case of RTK, that is receptor tyrosine kinases, which is the second class of receptor molecules, lipid hydrolysis is not mediated by the method which is shown in GPCR. Instead, it is mediated by recruitment of another phospholipase, PLCG. The PLCG is recruited to tyrosine phosphorylated proteins at the plasma membrane, that is the cytoplasmic leaflet of the plasma membrane, that is tyrosine phosphorylation of PLC-G by RTK. It is able to also stimulate the activity directly. So there are two methodologies, either it is activated directly or it is activated by recruitment of phospholipase C to tyrosine phosphorylated proteins. So this particular panel shows how exactly the formation of DAG and IP3 occurs, and we can see here how P1 is acted upon by kinase, which leads to phosphorylation leading to formation of PIP, which again is acted upon by kinase leading to formation of PIP2, which is then acted upon by enzyme PLC beta leading to formation of DAG, which remains associated with the membrane, and IP3, which is a soluble molecule which is released into the cytoplasm and it acts on specific organelles. Now IP3, that is inositol 1,4,5 trisphosphate, it acts as a second messenger. If the lipid cleaved is PIP2, then water soluble IP3 is produced, which is capable of rapid diffusion throughout the interior of the cell. These molecules, which are formed at the membrane, they diffuse into the cytosol and bind to specific receptors, IP3 receptor, which are located on smooth endoplasmic reticulum and other organelles, and after the binding it leads to release of calcium ions from the smooth endoplasmic reticulum. So IP3 receptors which are present on SER, they bind to the IP3 formed by the specific process of specific earlier process, and it leads to release of calcium ion. This can be, IP3 can be further modified to yield additionally phosphorylated phosphoinositols, which include diphosphoryl inositol phosphate. The IP3 receptor, it itself also functions as a tetrameric calcium ion channel. When IP3 binds to it, calcium ions start diffusing from the SER, that is smooth endoplasmic reticulum, into the cytoplasm. Now calcium ions, again they bind to specific target molecules and they initiate specific responses, some of which are contraction of smooth muscle cells and exocytosis of histamine containing secretory granules in a mast cell. These both activities happen when calcium ion level increase whenever there is binding of IP3 to SER. Inositol 1,3,4,5 tetrakis phosphate, that is termed as IP4, inositol 1,2,3,4,5,6 has hexaphosphate, that is termed as IP6. These are different products of IP3, and they activate specific channels, like IP4 activates chloride channel, IP6 is found in plants, in yeast also, and also in other animal cells. And this IP6 is gaining as interest as a potential anti-cancer agent because it promotes apoptosis in the cells. Now another second messenger which is formed is DAG, that is diacyl glycerol. Diacyl glycerol, contrary to the IP3, it remains bound to the inner leaflet of the plasma membrane. It recruits some certain other proteins, the primary of which is protein kinase C, PKC, which is also known as, and this particular PKC is recruited to DAG because it bears a specific domain which recognizes DAG, and this domain is termed as C1 domain. Now this PKC is able to phosphorylate a large number of other kinases like serine and threonine kinases, which set up in motion another set of reactions or another set of pathways. PKC represents a family of 9 genes which are grouped into 3 families. Apart from PKC, DAG also activates certain other proteins which have C1 domain, and these proteins are namely RAS, gastrin releasing polypeptides, DAG kinase and N-chimerin, and there are a number of other such proteins. Now this protein kinase C, as the name suggests, it is a kinase because it can add phosphate group to specific amino acids in the target protein. Now there are 3 types of these protein kinase C, namely conventional PKC isozymes, which require presence of both calcium, which is sensed by C2 domain, and DAG, which is sensed by C1 domain for activation. That is, it requires presence of both DAG and calcium ion, and thus it can only transduce signal which trigger PIP2 hydrolysis, because then only the calcium ion can be generated, because PIP2 hydrolysis only releases IP3, which can lead to release of calcium ion from the SER. This particular set of enzyme cannot be activated by processes which hydrolyze PC. Now there is another class which is novel isozymes of PKC which do not have a calcium sensor, but they have DAG sensor. So they have affinity of DAG which is 2 order higher than the conventional PKC isozymes, and they can transduce signals triggered by PC hydrolysis, that is phosphatidylcholine hydrolysis. And the third class of PKC isozymes are there, which do not recognize both DAG and calcium, which do not respond to both of them. Now, the signaling by conventional and novel PKC isozymes is terminated whenever there is phosphorylation of DAG by DAG kinase. As soon as DAG kinase acts on DAG and phosphorylate it, it cannot act as a messenger. It cannot activate the downward pathway. PKC isozymes are constitutively phosphorylated, but they do not act when they are phosphorylated. That means the phosphorylated condition is not sufficient for them to be active. They have to be activated by a certain other mechanism, which is DAG or calcium dependent translocation of these isozymes to the membrane. They are constitutively phosphorylated only when they are bound by DAG or DAG and calcium ion, then only they will be moving to the membrane, and then only they will be getting activated. So, phosphorylation does not automatically activate these enzymes. In cells which are not stimulated, PKC localizes to the cytosol where it is concentrated at specific regions, but PIPT hydrolysis provides calcium, which binds to the C2 domain of these enzymes, and it recruits the PKC to the plasma membrane. Once it is recruited to the plasma membrane, it binds DAG also, and once it is bound to DAG, these enzymes are activated. Binding of both the C2 and C1 domains to the membrane, it provides the energy to the PKC to release the autoinhibitory portion present in that enzyme and basically expose the active part of the enzyme. Once that is done, this PKC gets activated, and it can add phosphate group to the enzymes which are its own target. Protein kinase C isoforms have a number of important roles in cellular growth and differentiation, cellular metabolism, cell death, and immune responses. Cells have been engineered which constitutively express protein kinase C. That means they express protein kinase C continuously, and they exhibit a permanent malignant phenotype. That means they are cancerous cells, and they can also cause tumors in susceptible mice. So, what exactly this suggests, that this particular enzyme, that is protein kinase C, is very important in ensuring that there is no malignancy or there is no cancerous growth in the cells, because it controls the growth. If it is constitutively expressed, it cannot control the growth of the cell anymore. Now, apart from acting like this, it can also lead to termination of certain secondary messenger, certain signal transduction pathway. For example, it can also lead to phosphorylation of receptors like EGF receptors and insulin receptor. Once this happens, once the EGF receptor is phosphorylated or once insulin receptor is phosphorylated, they are internalized and destroyed. Once they are internalized, that means the response they were generating, it is stopped altogether. So, PKC, apart from activating certain pathways, it can also act as a terminator of certain specific signal transduction pathway. Now, these as I have suggested, there are specific proteins which bind to the phospholipid present in the membrane, and these protein recognize these particular phospholipids in the membrane by virtue of specific domains present in them. And there are several domains, namely pH domain, PX domain, and FYVE domain, which are found in the proteins which bind to these phospholipids. Binding of a protein by its pH domain to PIP2 or PIP3 recruits the protein to the cytoplasmic side of the membrane, and where it is able to interact with number of membrane bound proteins, which include activators, inhibitors, or other substrates. So, the protein which is going to recognize specific phospholipid on the membrane, it has to have certain domain like pH domain, which can go and bind or which can go and recognize these phospholipid in the membrane. Another important molecule which acts as a second messenger, as I have told already, is PIP3 molecule. Now, PIP2, it can be phosphorylated by PI3 kinase, which leads to formation of PIP3 molecule. PI3K enzymes involved in this particular process can be controlled by a large number of extracellular molecules, and the activity of PI3K has been associated with a large number of cancers in humans. Now, binding of growth factors to receptor tyrosine kinases result in the activation of PI3K isoform, which catalyzes the phosphorylation of PIP2 at 3' position to PIP3 in the plasma membrane. Though the PIP3 forms a very small percentage of the phospholipid present in the membrane, still once it is formed, the active PIP3 can lead to transduction of a great number of growth factor signals. Now this PI3K is also able to phosphorylate phosphatidylinositol and phosphatidylinositol 4-phosphate at 3' position to generate 3' phosphoinositide that also serve as second messenger. So one molecule of PIP3 can be activated or can be modulated in number of ways, can be transformed in number of ways, and all these transformed molecules can act as second messengers. The best defined target of PIP3 has been found. It is a specific kinase termed as AKT, which is a pro-survival kinase, that is the kinase which helps the cell to survive, and it is present in the cytosol of the unstimulated cell. Now PIP3, which is produced by PI3K activation, it recruits AKT to the plasma membrane and it leads to phosphorylation of the AKT molecule, which leads to its activation and downstream signaling. Once phosphorylated, AKT is then locked in an active conformation and it is released, and it can transduce its signal, and it can even activate genes which are present inside the nucleus. The signaling of PIP3 is terminated whenever there is dephosphorylation of PIP3 or dephosphorylation of AKT, and this particular dephosphorylation of PIP3 is catalyzed by a tumor suppressor PTEN protein, and dephosphorylation of AKT sites is catalyzed by tumor suppressor PHLPP proteins. So this particular panel shows how PI3K can lead to formation of PIP3, which can lead to activation of AKT, which can lead to activation of the downward signals or signal transduction. Now another class of molecules of phospholipids are sphingolipids, which are a class of bioactive lipids, and they also play a role in number of cellular responses. Ceramide is the one which is able to propagate information in stress response pathway, and it is formed by activation of receptors by stimulus such as cytokines or other ligands, or by non-receptor response to radiation, cytotoxic insult or infection by pathogen. Sphingosine, which is formed by cleaving of the sphingomyelin, it also has apoptotic roles, whereas sphingosine 1-phosphate, it is the one which is having pro-survival activity. Then there are classes of lysolipids, prostaglandins and other eicosanoids, which also play an important role in the transduction of signals. Now coming to ions as second messengers, ions are also very important in transmitting signals from outside to inside, and the most important ions are calcium and magnesium. These ions can be released into the interior of the cell through from outside to inside or from specific organelles present inside the cell, as we know is the case of calcium ion, which is present in smooth endoplasmic reticulum. This particular panel shows how Ca2 plus and Mg2 plus can activate a large number of pathways within the cell. Now these particular ions are released whenever there is a specific signal, and cells spend a large amount of energy in maintaining specific gradient. So whenever a signal comes to the cell, this gradient is crossed, and by the virtue of the electrochemical gradients, the ions are released into the cytosol, and they go to their target protein and they activate specific pathways. For example, the concentration of the calcium ion increases from 100 nanomolar to 10 micromolar within milliseconds in the cell whenever there is a specific signal arriving to that particular cell. And these calcium ions, they basically go and bind to specific proteins like synaptotagmin and troponin C, because these proteins have specific receptors for them and they activate specific pathway within that particular cell. And then there are ions like magnesium ions, which also play an important role. It has been suggested that they play a role contrary to the calcium ions. Whatever pathway the calcium ion is propagating, magnesium ion tend to inhibit that particular pathway. So in this way, calcium and magnesium ions both play an important role in cell signaling. Thank you.
With this, thank you. So thank you so very much for giving us this session. Friends, you are requested to be with us as we are back after a short break. Thank you. Hello friends, welcome back to this session. Friends, we welcome you in the another session where we are going to discuss more on cell signaling, and you have gathered lots of information on cell signaling through our previous session too, and as promised, here we are again. So for the discussion on cell signaling, we have with us in our studios Dr. Varunendra Singh Rawat. Dr. Varunendra Singh Rawat is Assistant Professor in Department of Zoology, and he is teaching in Hindu College. So let's welcome our guest Dr. Rawat, and let's try to understand more about the topic. Hello sir, welcome back to the session.
So friends, I'll be talking again about an important aspect of cell signaling. We have been talking about cell signaling in detail, and in this particular lecture I'll be discussing another important aspect of cell signaling, which deals with RAS-MAP kinase pathway and the signaling by insulin receptor. We have been talking a lot about different types of receptors, how they perceive the signal, how they bind the signal which comes to them. And after binding signal, how exactly they modify certain other proteins, what exactly is the mode of modification of other proteins which leads to signal transduction from outside to inside. So there are different types of receptors as I've already been told, like GPCRs and RTK. So we have discussed GPCR in detail, and we have also discussed RTK in detail, which is receptor tyrosine kinase. Once the signal molecules bind to receptor tyrosine kinase, the downward pathway of that particular binding of messenger molecule to the receptor is RAS-MAP kinase pathway, which I'll be talking about in detail in this particular topic. And then there is insulin receptor, which exactly how exactly that works, which I'll be talking about. It is a type of RTK, that is receptor tyrosine kinase, and how does it differ from other RTKs, and what exactly is the mode of working of this particular receptor. I'll be talking about that. So the learning objective and the outline for this particular topic would be basically description of RAS-MAP kinase cascade and signaling by insulin receptor. How exactly signal transduction occurs through RAS-MAP kinase cascade, and what exactly is the mode of functioning of insulin receptor in the cell. Before going into the RAS-MAP kinase, let's just recapitulate what exactly RTKs were. Receptor tyrosine kinase are a type of receptors present on the cells, and they are able to bind whatever signal they are able to perceive, and once they bind the signal, the tyrosine kinase activity of these particular receptors is activated. Now what exactly are kinases? Kinases are those molecules or enzymes which can phosphorylate, that is, they can add phosphate to specific amino acid in protein. So receptor tyrosine kinase are special because they can autophosphorylate, that means they can lead to self-phosphorylation or addition of specific phosphate group to their own tyrosine residue. So coming to receptor tyrosine kinases, whenever they perceive the signal, they dimerize. That means these are present in two copies, and when they bind the ligand, they are able to form a dimer, and the cytoplasmic tails of these dimers, they are able to phosphorylate tyrosine residues of the adjacent monomer. So two molecules come together, the kinase activity of one monomer is able to phosphorylate tyrosine residues of the other monomer, and vice versa. So the end result is that the tyrosine amino acids in the dimer get phosphorylated, and therefore these receptors get activated. So the essential part is binding of ligand leads to conformational change in the receptor, which leads to activity of the kinase, which leads to its own activation. But there are a small number of RTKs, like I talked about the insulin receptor and IGF-1 receptors, which are present as inactive dimer. That means they are present as dimer by themselves. They are present as dimer. They are not present as monomers. All the other type of RTKs are present as monomers, but once the ligand binds, they become dimer. But in this particular case, like insulin receptor, they are present as dimers, but they are inactive. They become active only when the ligand binds. So the dimerization is prior to binding of ligand binding, but once the ligand binds, it gets activated. Whereas in other RTKs, dimerization occurs only after ligand binding, and it follows activation of the RTK. Now for most RTKs, receptor dimerization results in the juxtapositioning of two protein tyrosine kinases domain, as I have already told, and this leads to trans-autophosphorylation, wherein the kinase activity of one monomer phosphorylates the tyrosine residues of the other monomer and vice versa. This particular panel shows how it exactly happens. The ligand can bind and it leads to dimerization of the two monomers, which lead to conformational change in the cytoplasmic tails of the monomers, which leads to trans-autophosphorylation. Once they are phosphorylated, the cytoplasmic tails are recognized by certain other proteins, which could be kinases, by the virtue of presence of SH2 domain or PTP domain in them. That means there are specific domains present in the protein, target protein, which go and bind to the phosphorylated tails of the RTKs, that is receptor tyrosine kinases. There are also a class of non-receptor tyrosine kinases which are present in the cytosol, but we are talking about receptor tyrosine kinases. Now, once they are activated, their kinase activity gets activated, it serves as a binding site for a large number of cytoplasmic signaling molecules, and these proteins are able to associate with other proteins, and they basically activate a set of a chain of proteins, which leads to transduction of specific signal which was being carried by the original messengers. Now, the pathway which follows the RTK, once the RTK proteins have been activated, the receptors have been activated, they can activate one of the pathway, which is RAS-MAP kinase pathway or RAS-MAP kinase cascade. It is termed as cascade because it amplifies the signal. Now, this particular set of pathways activated once the RTK pathway or the receptor tyrosine kinase get activated, and the genes involved here are RAS genes. Now, these RAS genes they are very important genes, because about 30% of all human cancers which have been recorded in humans, the cancers which have been recorded in humans, they contain mutant versions of RAS genes. So, there are a large number of RAS genes, and wherever these genes are mutated, they might lead to development of cancers in the individuals. So 30% of all human cancer, that's a very large percentage, they are related to RAS gene. Now, this RAS gene was originally described as a product of a retroviral oncogene, but later on it was found that it is also present in the mammalian host. Earlier it was found that this RAS gene was present in a retrovirus and it was an oncogene, but later on, after genomic studies, it was found that this gene is also present in the genome of the humans, and once it gets mutated, it will lead to formation of cancer in that particular individual where that gene has got mutated. Now, these RAS proteins are a part of a superfamily of more than 150 small G proteins. Again, these are type of G proteins, like in G protein coupled receptors we had heterotrimeric G proteins, but these RAS are not heterotrimeric, they are monomeric, and they are small in size because they are monomeric, but they are G proteins. That means they can add GTP or GTP, they can lead to addition of GTP or GTP in them, and these include a large number of proteins. This particular family, like RABS, SAR1 and RAN, all of these are involved in cancer development in mammals. Now, all these proteins are involved in regulation of numerous other processes like cell division, differentiation, gene expression, cytoskeletal organization, vesicle trafficking, and nucleocytoplasmic transport. So, this particular family is very important, not only because it when mutated it can lead to cancer development, but apart from that, when it is normal, it leads to gives rise to protein which take part in a large number of processes which are required for a normal cell to function. Now, this RAS is a small GTPase. As I have already told, it is a G protein. It is a GTPase which is present at the inner surface of the cytoplasmic of the plasma membrane. That is, the cytoplasmic leaflet of the plasma membrane has bound RAS protein to it. It is attached covalently by a lipid group which is embedded in the inner leaflet of the bilayer. Now, this RAS, it is a G protein. It is functionally similar to G proteins which have been talked about in G protein coupled receptor, but the difference is that it consists of only a single small subunit. Now, these proteins, as termed earlier, these are G proteins, they are GTPase. They can either be bound to GTP or they can be bound to GDP. Once they are bound to GTP, they are in active state. Once they are bound to GDP, they are in an inactive state. So, they can exchange their GTP with GDP and GDP with GTP. Once they are bound to GTP, they become active, and once they are bound to GDP, they become inactive. Now, this RAS GTP, which is an activated form, it activate the downstream signaling process, and it can be turned off. This signaling can be turned off when the GTP is hydrolyzed and phosphate group is released, leading to formation of GDP. The mutant RAS protein which has bound GTP but which cannot be hydrolyzed. There are certain mutant forms of RAS which can bind GTP, but in them the GTPase activity is not there, so GTP cannot be hydrolyzed, so they are constitutively active, that is they are in on position and they keep the cell in continuously proliferative mode, which can lead to development of cancerous cells wherein there is no growth control and no divisional control. Now there are certain set of proteins which take part in changing the active form to inactive form and inactive form to active form, which ensures that the RAS is activated at specific point of time and only when required, and it gets inactivated when not required. So the cycling of RAS between these active and inactive state is aided by certain accessory proteins which are able to bind to this particular RAS protein and regulate its activity, and these proteins, these class of proteins are GTPase activating proteins GAPS and guanine nucleotide exchange factor GEFS and guanine nucleotide dissociation inhibitor GDIs. As the name suggests, GAPS are the proteins which will activate the GTPase activity, which will lead to hydrolysis of GTP. That means they help in converting the active form to inactive form. Then we have guanine nucleotide exchange factor. As the name suggests, these help in exchanging the nucleotide factor, that is exchange of GDP with GTP, which suggests that they are the ones responsible for activating the protein. Then we have guanine nucleotide dissociation inhibitor. As the name suggests, they are inhibitors to dissociation of GDP. That means they help in keeping the RAS protein in inactive form. So an interplay of these three proteins decides whether the RAS becomes active or inactive. So talking about them in detail, GAPS, that is GTPase activating protein, they stimulate the hydrolysis of bound GTP, which inactivates the G protein, as I have already told. Thus it shortens the duration of G protein mediated response, because these are the protein which are responsible for hydrolysis of GTP to GDP. They will shorten the response whenever they hydrolyze the GTP. Now if there is a mutation in RAS GAP gene, what happens then? If there is a mutation in RAS GAP gene like NF1, it can cause cancers. Like whenever there is mutation in NF1, which is the RAS GAP gene, it causes neurofibromatosis 1, which is a disease in which patients develop large numbers of benign tumors which are termed as neurofibromas along the sheaths that line the nerve trunks, and these can be seen as globule protruding from the skin of the individual. And this happens because of mutation in RAS GAP gene, because the RAS protein cannot be inactivated anymore because the GTPase activity is not there in this particular protein. What about guanine nucleotide exchange factors like GEF? These are the protein which bind to inactive monomeric G protein. Which form is inactive? The form which is bound to GTP. Now these GEFs bind to inactive protein. They stimulate the dissociation of the GDP. Once the GDP is dissociated, it is released from the G protein, and in its place comes another protein which is GTP, and once GTP is bound, it leads to activation of G protein. So GEFs are the one which activate this particular RAS protein. Now coming to the third class, which is guanine nucleotide dissociation inhibitor, GDIs. What do GDIs do? They inhibit the release of a bound GDP from a monomeric G protein, and it maintains the protein in inactive GDP bound state. So what exactly they are doing? They are binding to the RAS protein which already has GDP bound to it. Once they are bound, there is no possibility of GDP getting released, and because there is no possibility of GDP getting released, there is no possibility of GTP coming in and binding to that particular site, and because GTP cannot bind, that means protein will remain in inactive state. What happens if there is a mutation in these particular proteins? If a GDI is formed, which though it binds to the RAS protein, it is not able to dissociate ever, what will happen then? Then the RAS proteins will never get activated, and that will be having serious consequences for the growth and division of the cells wherein these particular genes are mutated. Now, this particular panel shows a summary of how exactly these three classes of proteins take part in RAS MAP kinase in the activating the RAS protein. We can see here there are GDI, GEFs and GAPs. So, GAPs are the ones which will lead to hydrolysis of the GTP to GDP. Once that is done, the RAS proteins gets inactivated and the target protein cannot be activated anymore. Once there is bound GTP to the RAS protein, it can only be exchanged once the inhibitor is released. GDI goes and binds to RAS protein which is bound to GTP. Once GDI is bound, the protein cannot be activated, but once GDI is released, then there is a chance for GEF, that is guanine nucleotide exchange factor, to come and release GDP from the RAS protein, and in its place will come GTP, and then the RAS protein gets activated and it can activate the target protein by its kinase activity. So, there is a fine interplay between these three classes of proteins which ensures that there is a timely activation of RAS protein and timely inactivation. If any of these switches or any of these proteins get mutated or inactivated, it will be having disastrous consequences for the cell wherein this particular mutation occurs. Once the RAS protein gets activated, it leads to activation of the cascade, which leads to activation of a large number of the intracellular signals. And this particular RAS MAP kinase cascade is basically activated by a large number of extracellular signals, and it plays a key role in regulating vital activities such as cell proliferation and differentiation. This pathway relays the whatever extracellular signal comes from outside, it relays from the plasma membrane to the cytoplasm and then into the nucleus, so that specific genes get activated and specific signal are transduced in effective manner. Now, when this pathway gets activated by a growth factor like EGF, that is epidermal growth factor, or PDGF, that is platelet derived growth factor, these factors basically go and bind to receptor tyrosine kinase. As I have already told that this pathway comes after the receptor tyrosine kinase gets activated. Now, these RTKs, once they are activated, they possess phosphorylated tyrosine residues which act as docking site or the binding sites for the adapter proteins like GRB2. These RTKs have sites which are recognized by adapter proteins like GRB2. These GRB2 bind to SOS, which is a guanine nucleotide exchange factor for RAS. Now, here we can see there are a number of proteins involved. RTKs are getting activated, they are binding GRB2. GRB2 in turn is binding to SOS, which is a protein which acts as a GEF for RAS proteins. Now, creation of a GRB2 binding site on an activated RTK receptor, it promotes translocation of GRB2 SOS complex from the cytoplasm to the cytoplasmic surface of the plasma membrane, which leads to placing of SOS in the close proximity to RAS. Now, RAS, as I have already told, is present in the inner leaflet of the plasma membrane. Once the RTKs are activated, the GRB2 binding site on the receptor, it promotes it. Once this receptor is activated, it promotes binding of the GRB2, but this GRB2 does not bind in isolation. It brings with itself SOS, which is a nucleotide exchange factor for RAS. Now, when this complex goes and binds to the receptor tyrosine kinase, then it basically SOS leads to activation of RAS, because SOS is a exchange factor, nucleotide exchange factor. It leads to exchange of GDP with GTP in the RAS. Once RAS, that is RAS, it is bound to GTP, it gets activated, and then it can activate a large number of signaling proteins like RAF protein. Now, once the RAS is activated, it can activate other proteins by its kinase activity. Now, one of these proteins is RAF. Now, this RAF is a serine threonine protein kinase. That means it goes and activates serine and it adds phosphate group to serine and threonine amino acids. Now, once this RAF is activated, it can activate another protein kinase MEK, which is activated which is activated by phosphorylation. Now once MEK is activated, it phosphorylates other proteins like ERK1 and ERK2, which are MAP kinases. So these are other set of kinases. So what we are seeing here is that there is a chain of kinases taking part in this particular cascade. One kinase is activating another kinase by phosphorylating it, and then that kinase is again activating another kinase by adding phosphate group to that particular kinase. So MAP kinase is the end like ERK1 and ERK2 are the MAP kinases, which are like end of the kinase chain, and they activate other proteins like transcription factors, protein kinases, cytoskeletal proteins, apoptotic regulators, and other signaling proteins. Once activated, the MAP kinase, once ERK1 and ERK2 are activated, they are able to move inside the nucleus, where they phosphorylate and activate a number of other transcription factors and other nuclear proteins. Now these transcriptor factions are the ones which activate a certain set of genes. Now this pathway lead to activation of genes because transcription factors are activated. They lead to activation of genes which are involved in cell proliferation, and these genes include cyclin D1, which is a very important gene which drives the cell from G1 into S phase. Now many of the proteins which play part in this particular RAS signaling pathway were discovered because they were encoded by cancer causing oncogenes. RAS, RAF and transcription factors like FOS and JUN are generated at the end of the pathway, and genes for several of these RTKs are situated at the beginning of the pathway, including the receptors for both EGF and PDGF, and these have been identified as oncogenes. In yeast, this particular cascade is required for cells to respond to mating factors. In fruit fly, it is recognized. It's required for differentiation of photoreceptors. In flowering plants, it is required for initiating a defense response. Now, in each of these cases, there is a core of the pathway which contains enzymes which act sequentially, as I've already told. There is a kinase, MAP kinase, MAPKK, and there are about 14 such kinases found in mammals. MAP kinase, that is MAPKK, 7 such MAPKK are found in mammals. These MAPKKs, they can phosphorylate tyrosine residues as well as serine and threonine residues in their target proteins. And what are their target proteins? Their target proteins are MAP kinase, that is MAPK. There are about 13 such MAPK found in mammals, and these MAPKs have a tripeptide near the catalytic site which contains sequence threonine X tyrosine. That means these threonine in the active site can be activated by MAPKK. These tyrosine can also be activated by MAPKK. So there is a set of kinases which act sequentially to transduce signal which has come from outside. And by utilizing different members of these protein families, these mammals are able to assemble a number of different MAPKs kinase pathway and transmit a number of different signals in response to different type of extracellular signal. Now when cells are exposed to damaging chemicals, the signals are transmitted through these pathways which tells the cell to withdraw from the cell cycle and ask it to not divide any further, because if a mutated cell divides, then it will lead to serious problems. This particular panel shows how RAS MAP kinase involves a large number of a sequential action of MAPKK, MAPKK and MAPK, and how it activates certain transcription factors and how then specific genes get activated. Now coming to signaling by insulin receptor, as we know that all the mammals and humans included, they maintain a specific level of glucose in their blood. Whenever there is a downfall in the level of glucose in the blood, certain signals are released which cause the liver to break down glycogen and release glucose in the blood. And whenever there is ample amount of glucose present in the bloodstream, it causes release of certain signals which lead to uptake of glucose through the bloodstream into the liver, so that glycogen can be formed. So there is a specific interplay of signals here. We know that whenever there is low level of glucose, then the alpha cell of pancreas secrete a hormone which is termed as glucagon, which act through GPCR and stimulate the breakdown of glycogen. But whenever the glucose level rises, beta cells release insulin, which informs the cell that there is a large amount of glucose in the bloodstream, and this leads to uptake of glucose by the cells, and this glucose can be utilized to form glycogen. So there is specific interplay of the hormones, of the signals and their receptors. Now each insulin receptor is composed of alpha and beta chain, which is derived from a single polypeptide. Now the alpha part is completely extracellular in its orientation. The beta part has extracellular domain, intermembrane domain and cytoplasmic domain. but they both alpha chain and beta chain are linked together by disulfide bonds with each other. Now these alpha and beta heterodimers are held together by disulfide bond, as I've already told, and they even when they are inactive, they occur in dimeric form. They are present in dimeric form, and whenever they receive the signal, they become activated. Now we can see in this particular panel there is alpha chain which is present outside the cell, beta chain which has extracellular domain, intermembrane domain and cytoplasmic domain, and two such alpha beta complexes form one dimer, which is always present in dimeric form even when it is inactive. Once a ligand like insulin comes, then it binds to both the binding site in the dimer, and it activates the cytoplasmic tail of the beta chain, which leads to phosphorylation of the tyrosine residues of the two cytoplasmic tails of the two beta chains, because it is a type of RTK only, receptor tyrosine kinase. So there are several tyrosine sites which can be phosphorylated, and three of these sites are present in the activation loop. In the unphosphorylated state, the activation loop has a conformation wherein it cannot be activated, but after phosphorylation of the tyrosine residue, the activation loop assumes a new conformation which helps in activating certain other set of enzymes, because this conformation brings in residues closer to the catalytic site. Now most RTKs possess autophosphorylation site, as we already know, but insulin receptor is an exception because it associated with a small family of docking proteins termed as insulin receptor substrate, IRS substrates, and these IRS provide binding site for SH2 domain of the signaling protein. So this receptor bind to IRS proteins, which then bind to signaling protein which have SH2 domains. After binding of the ligand and kinase activation, there is autophosphorylation and activation of specific domains like PTB domain, and it leads to activation of signaling molecules. Four family of IRS have been identified, and of these IRS1 and IRS2 are the most relevant, and these families, they basically bind the receptor through pH domain. This particular panel shows how the insulin dimer bind to specific proteins and leads to activation of certain other proteins in the pathway. Now these IRS1 and 2, when they bind to the receptor, they phosphorylated tyrosine residues on these docking proteins themselves, and then they phosphorylate a large number of proteins like PI3 kinase, GRB2 and SHP2, which activate the downstream signaling pathway. One of the important protein which gets activated is PI3K, which consists of two subunits, and once it is activated, it leads to formation of PIP2 and PIP3, which remain in the cytosolic side of the plasma membrane, and they activate the other protein which fall down in pathway like PKB protein, which is also termed as AKT protein, and it leads to activity of certain other protein in the pathway. Now this PKB is involved in regulating glucose transport, and glucose transporter GLUT4, which carries out insulin dependent glucose transport from the blood, is also responsible for maintaining the glucose levels. However, in the absence of insulin, GLUT4 is present in membrane vesicles in the plasma, in the insulin responsive cells, and these then fuse with the plasma membrane, and it leads to uptake of glucose. Now there is an interplay of GLUT4 protein and specific PI3 kinase protein, which leads to fine balance of glucose within the cell, and it ensures that cell has ample supply of glucose whenever required, and this is how the insulin receptor helps in maintaining the glucose level in the cells. Thank you.
With this note, thank you so very much for giving us this session. Friends, you are requested to write to us at info.cec@nic.in if you have any queries or feedback for this particular lecture. We are going to meet again very soon and would be discussing more. Till then, take care. Goodbye. Thank you, sir. Thank you once again.